Let's all wait for few more minutes, like 5 more minutes for others to join, then we'll start this session. Hello, good afternoon all. I'm audible to everyone. Okay, so we'll start the session. Welcome you all to today's tutorial session, which will basically be summing up or summarizing the whole week 3 lecture videos or lecture content, and then we'll try to look at few of the questions based on whatever we have summarized. Okay. And also again in between, I request you in case someone's wanting to join, because once I share my screen and start presenting, I am unable to look at who is joining or waiting to get inside the call. So just let me know, unmute yourself and just let me know if someone's waiting. Okay. So we'll start the session. Uh, is the presentation visible to everyone? Oh, I haven't shared the screen only. Sorry. Now is it visible? Okay. Sorry, I didn't share my screen. Okay. Okay. So basically this week and I think the next week lecture videos are all about, how to genetically modify an organism or how to introduce genetic changes with respect to what we desire or with respect to our needs. Okay. So now, genetically modified organism is basically any kind of organism whose genetic makeup or genetic constituents have been manipulated according to certain whatever requirement that may be, whether it's experimental, whether it's diagnostic purpose or therapeutic purpose, and any organisms, mostly bacteria, plants, animals, or fungi like yeasts, viruses, all of these we can introduce uh, synthetically certain kind of genes of our interest, and then these organisms, this these genes, even though it's foreign to these organisms, or these are not naturally present in these organisms, they can still, we can make these organisms, we can manipulate these organisms to make or code for the genes, or code for the proteins of our interest, or code for any or basically it doesn't have to be proteins. We can also stop at the RNA level as well. Okay. So this is a basic outline of what happens in a genetically modified organism. Suppose uh, we want to, save the crop plants from insects. So what happens is some sort of gene that is harmful for the insects or that kills the certain kind of insect that or infests the plants or crop plants. That kind of gene, that protein encoding gene, that insecticide encoding gene can be created using recombinant DNA technology. What happens is these are certain carriers which we call plasmid or vectors, or simply we can tell them as vectors, which are used to carry a certain sequence of our interest or genetic sequence of our interest from one organism to another. Okay, or in the same organism, the strains can be different. All right. So what we basically do is, this is the genetic sequence, and we chop it off using certain kinds of enzymes, which are the restriction enzymes. Okay. And these are the chopped off pieces, and we chop it off ideally with the same set of restriction enzymes, these vectors, as well as the gene of our interest. Okay. Now these fragments, these chopped off genetic sequence are then joined or ligated to our plasmid vectors, and thereby creating, see, this recombinant plasmid. And then this is then again put inside an organism of our interest, whether it's, bacterium, yeast, or plant cell. Uh, then that is then allowed to grow. In this case, in this example, this has been put into these recombinant genetic sequence has been put into plant cells, and these plant cells are grown, and which ideally, so here in this example, this green colored one is an insecticide coding encoding gene. Okay, so these plant cells, some of them might take up the like this particular recombinant vector, some might not take the recombinant vector. So I think that in the previous classes we saw that whatever, so we use a selective media. Ideally, generally, a selective media is used, wherein suppose this vector has a certain antibiotic resistance gene, which is generally or naturally not present in the plant cells. Then what will happen is only those cells will grow which have taken up these recombinant vectors. Okay. So this is what is meant by selection. Select for insecticidal cells, and then this is again transfected into plants, and plants are propagated. Then what will happen? That insecticide chemical, whatever is that chemical or protein, that will be encoded by the plants. And then when suppose these insects feed on the plants, they will be, uh, either either they will be harmed in some way or killed directly. Okay. Now what? So the very first genetically modified organism was created by two biochemists, Boyer and Cohen. So what they did for the very first time, they inserted a kanamycin-resistant gene from one bacterium into a plasmid of another bacterium. This particular bacterium, this bacteria number 2, doesn't really have kanamycin resistance. Okay. So what happened when they inserted this particular gene into the bacterium number 2, the number 2 bacterium became kanamycin resistant. So this was the first genetically modified organism, which GMO is the short form for genetically modified organism, which was the first GMO developed. And these, they again, Boyer and Cohen, they transferred a gene from xenopus, which are actually frog or toad species, into a bacterium. This was again the first genetically modified organism with genes inserted from one kingdom to the other, right? Because bacteria is completely prokaryotic and toads are completely eukaryotic and much higher level of eukaryotes as compared to prokaryotes. Okay. And then during this time, later there was this company, Gene Tech, which uh, made recombinant human insulin and then made E. coli species, modified E. coli bacteria to produce human insulin. This was termed as humulin. And then I think after around 4 to 5 years, FDA approved the use of humulin, which was the first product from any genetically modified organism to be used for consumption, mainly for our consumption or for human consumption. Now, this is taken from the lecture videos only. So how can we really, what are the possible approaches for isolating gene of interest? There are two scenarios when we know what our gene of sequence, what the genetic sequence of our interest is, and we this scenario, and when we don't really know what is the suppose we got a completely new organism and we want to study that organism, we don't really know what its genetic sequence is, or we found a certain characteristic in an organism, we don't know what particular genetic sequence encodes for that particular characteristic. Okay, so these are the two case scenarios. So when just briefly we will go through each of the cases, what happens and what needs to be done. And so the first thing is when the gene sequence is normal. Okay. So basically, what does cloning mean? Like the cloning word, we are like, okay, we are cloning this particular gene of interest. This what does cloning mean? It means to, produce multiple copies or identical copies of a particular gene of interest. Okay. In general, cloning of a gene refers to this particular thing that you are actually creating identical, many numbers, several copies, identical copies of a particular gene of interest. Okay. Now, so here, first, when the genetic sequence is not known, either when at the DNA level, what we can do is we can create a genomic library and then uh, target our particular gene of interest, or we can look at the transcriptome. Transcriptome is basically whatever amount of or the pool of RNA that has been transcribed or encoded by the DNA of our from the DNA of our genome. that is transcriptome, and that RNA can be reverse transcribed into DNA to form again, like genomic library. Here we have to form that complementary DNA library, and then further downstream process of screening, cloning the fragments, etc. will be taking place. So, how to really construct a genomic library? Uh, what the very first thing definitely you have to do is you have to isolate the genomic DNA, the whole sequence of nucleotides or um, genetic sequence has to be isolated from the organism of our interest. Okay, and then it has to be chopped off using certain enzymes. Okay, with having different kinds of fragments, different lengths of fragments. Now, usually what happens is genomic DNA, as compared to what we were looking at, the vectors we were talking about, right? Plasmid, uh, before, I mean, in the previous slides, we're talking about the plasmids. Now, plasmid DNA is much smaller than genomic DNA. Suppose E. coli, which is actually a bacteria, its genome is around, I think, around 4.5 or around 4 megabases long. So this is like huge as compared to plasmids, which are only around, ideally, around 3 to 5 kilobases long. That's megabases, and this is around in the region of kilobases. Okay, this particular genomic DNA, after digestion or after treating with certain enzymes, it will chop it off into fragments, which ideally will result in large chromosome fragments. Okay, so genomic DNA, basically we are talking about the chromosomal content of the organism of interest. And then after chopping it off, we purify the DNA fragments. And then what we saw in the previous slide, that we will joining these purified DNA fragments, these are the ones, and into a suitable vector. But we will come into these types. There are many kinds of vectors, into a suitable vector, and then that recombined vector will have to be introduced into an organism of our interest. Like in the previous example we saw, we introduced into plant cells. All right, then insecticide uh, coding gene, which was cloned into a plasmid, that recombinant plasmid was introduced into plant cells. Similarly, here uh, we have to ligate the fragments into a suitable vector and then transformed into this organism, thereby forming multiple copies of that particular genomic DNA, multiple fragments having these organisms are then stored, and this and these organisms have the multiple fragments, which are multiple clones of each fragment, thereby forming a genomic library. And so, okay. Now the step one is isolating genomic DNA. So we'll look into a little bit more details. Like these are all, I think you are all at the college level. So these will be part of your practicals as well, molecular biology practicals. So each step has a certain principle behind or theory behind, okay, why are we doing this particular step? What is the reason behind using a particular solution and not the other solution? Okay. So how to isolate genomic DNA? The very first thing will have to be done is you have to break open the cell in order to get your genomic DNA out of the cell. All right. So you have to break open the cell or lyse the cells in a suitable using a suitable buffer, which is commonly referred to as lysis buffer. Okay. Then because you need genomic DNA, but then when the cell is getting opened up, cell is getting lysed, you will have other biomolecules as well coming out, like proteins, RNA, polysaccharides, and other different organic molecules. You don't really need that. You are interested in getting pure genomic DNA. Okay. So again, that lysed contents have to be treated so as to get rid of the other unwanted components and only get pure genomic DNA. Okay. And then when the genomic DNA is obtained, it is precipitated and then finally analyzed on using electrophoresis gels, and also not only on gels. You very briefly I had discussed in the very first tutorial session about absorbance ratios. Hmm? So like there's genomic DNA, we get the value, we take the reading, absorbance reading at 260 nanometers wavelength. Okay. Now 260 nanometers wavelength, uh, nucleic acid absorption takes place, and at 280 nanometers, proteins or aromatic, basically aromatic amino acids show their absorbance. And at 230 nanometers, other organic solutes or polysaccharides, these give the reading at 230 nanometers. So along with running on gels, what also needs to be done when actually you are performing these things in laboratory, what you should be doing is you take the ratio of 8 to 6 absorbance at 260 nanometers divided by absorbance at 280 nanometers. Okay. So at 280 nanometers, then that ratio of 260 by 280 will give you an idea of the amount of protein contamination present. Okay. So for DNA, it ranges in between, the ideal ratio should be between 1.8 to 2. Okay. Similarly, A260 by 230 ratio, A260 by absorbance at 230 nanometers will give you an idea of the amount of contamination by other polysaccharides, organic solutes, etc., which will give you an idea, okay, how pure your purified genomic DNA is, or in fact at what step of this whole purification things might have gone wrong, that you are getting a contaminated sample that can be visible on the gel, or you are getting a not so good ratio of the absorbance ratios at 260, 280, and 230 nanometers. Okay. Again, at each step, we will go into little bit of details. Okay, one by one. Now, first is you have to break the cell open or lyse the cells. Now, for genomic DNA, because genomic DNA is such a huge thing, the chromosomal content is around 4 megabases long. So what happens is you can be a little like not really so gentle. It can be a little harsh. Also, the lysis. What happens is like first is like simple mortar and pestle, like in Indian homes you usually get that. Similarly, the cells can be lysed using a mortar and pestle. Or what happens is, suppose plant samples, okay, leaves or something like that, what happens is we put it in liquid nitrogen. Liquid nitrogen is at minus 192 degrees Celsius. Now it makes the whole thing so brittle, and then you simply put it inside, a mixer grinder. Then it will be completely powdered. So you have lysed the cells. Okay. The whole leaves that you see, the green leaves, suppose Arabidopsis, like that, it can be lysed in that way. Okay. So ideally, comparatively bulky samples can be lysed using a mortar and pestles. But what will happen is, after lysing these uh, bulky samples, the metabolites, if you're interested in certain metabolites or something, then that metabolites and samples can be extracted further by adding suitable solvents. Okay. Next is bead beating. What happens is people use glass or ceramic beads to crack open the cells. Okay. These are actually, see, mortar and pestle, bead beating. These are mechanical shearing kinds, or mechanical ways of slicing the cells. Okay. So what happens is you have very small, uh, which can actually like multiple tubes of maybe uh, like millimeter sized beads or spheres. These are put, multiple such beads are put inside, in a tube, and then you either centrifuge it or not centrifuge it. Usually there's a machine called vortex. What happens is, it once you just touching the thing, maybe I can show you what a vortex is. See, what happens is, this is kind of, this is an area where you put your tube. Okay, this black color thing, and this can regulate the speed. So this vibrates at a very high speed. So the moment you put your tube on that black area, the tube also vibrates very strongly. Okay. So you can regulate uh, that particular vibrations here using this regulator or knob here. And then see, and this can be different types also. So these ideally are the tubes I'm talking about. These can be smaller as well, and that particular vibration can cause can actually lyse the cells. Okay, use so the beads can be glass or ceramic in nature. Then there's sonication. This is the most commonly used lysis method for most types of you know whether it's protein purification or ideally for protein purification, sonication is definitely used. Like after mechanically lysing the cells or resuspending the cells in a lysis buffer, sonication is definitely the go-to method. What happens here is a titanium probe which can emit, vibrations. Okay, which are the whole setup is called sonicators. There's a certain kind of probe here which produce rapid vibrations, and this is actually immersed in your tube, which has uh, the cells suspended in a suitable buffer. Okay, I think in the previous class we talked about how to lyse the cells. There can be different kinds of treatment to cells, like different kinds of enzymatic treatment. We will look into the next slide also. So, such kind of solution where the cell is lysed and resuspended. In that, what happens is when the rapid vibrations are produced, it leads to cavitation. This process is called cavitation. What happens there is tiny bubbles are produced within the cell suspension. Okay, and these bubbles can explode. Okay, thereby producing local shock wave which can disrupt the cell wall and just lyse the whole thing. Okay, so this is actually like a bomb explosion kind of thing where things can scatter away and the cell contents can come out. Okay, then there are homogenizers, which is again a kind of mechanical method only, like bead ones, glass or ceramic beads. Again, the same thing. What happens here is it's a little bit more sophisticated kind of stuff, where what you do is uh, you put your cells that you want to break open. Those particular cells are passed through a certain kind of apparatus which is having an opening smaller or much less wide than what the dimension of a cell is. That means it is very small, and then when the cells are squeezed through a tube that is slightly smaller than the cell dimensions, what will happen is the protoplasm, ideally if we are talking here about the bacterial cell, suppose, then the protoplasm which is without the cell wall and the cytoplasm devoid of the cell wall, cytoplasm and plasma membrane devoid of the cell wall. So that can pass through, and like we put pressure, and then only the cell wall can remain outside, whereas the other stuff can get inside and pass through that particular apparatus. Okay, so this is what is written here. We can shear away the outer layer by using homogenizers. Okay. Then there's high temperature lysis also, like at high temperature and pressure, chemical bonds can break within the cell walls. And but then, at high temperatures, proteins can also get denatured. So, but high temperature lysis can be quick as well as dirty, and you can directly microwave or autoclave to generate the required amount of heat and pressure. But since it is denaturing proteins, now suppose you want to purify proteins, or like again, whatever your downstream goal is, requirement is, based on that, you should be using this high temperature lysis. Ideally, this should be avoided but when you're trying to purify samples which get denatured by heat, because this heat is like really high, like the heat generated is at very high temperatures. Otherwise, the cells, suppose bacterial organisms, there are many bacterial organisms that thrive in high temperature conditions. Then this won't really be a very good option to break open such kind of cells. Okay, then definitely there are enzymatic lysis which can break open cells, or can react with the components that make up the cell wall of the cells or membranes of the cells. Okay, so depending on the organism, one can use cellulases for plant cells, chitinases for organisms having chitin, fungi. Okay, bacteriolytic enzymes, there's lysozyme also. Okay, bacterial cell wall has peptidoglycans, right? So lysozyme is an enzyme which can destroy the peptidoglycan and thereby it can degrade the cell wall of bacterial cells. Then there's manganese or glycanase. Okay. Wherever glycanase means glycan moieties are degraded. Okay. So enzymatic lysis can be also useful in cases where we want to isolate the protoplast, a cell without the cell wall, without the cell wall. Okay, next comes chemical lysis. Now, there are different organic solvents like alcohols, ether, or chloroform. What these kinds of solvents do, it can disrupt the cell wall. It can make small pores in the cell walls, okay, as well as in the membranes. You know what happens, suppose when I think you guys have heard about transfer RNA molecules, right? tRNA, which are actually very small. These are around, I think, 77 nucleotides long, 77 to 80 nucleotides long. So such a small molecule, when we want to really, extract it out of, suppose bacterial cells, uh, people use chloroform to, you know, to make very minute pores. It can actually disrupt or make pores in the cell walls as well as in membranes so that the small molecules can come out. The other molecules like the genomic DNA, plasmids are also much like 5 kilo bases, whereas this is only 77 bases. So plasmids and all such other heavier or longer nucleotide molecules or heavier molecules or bigger like molecules having bigger dimensions can stay inside and smaller molecules like these tRNA, etc. can come out. Okay. So this is how chemical lysis using organic solvents can be useful. This is handy to extract hydrophobic molecules also in case of plant pigments. Also, there's ethylene diamine tetraacetic acid or EDTA, which is a chelating agent. Basically, it chelates away divalent ions. Okay, mainly divalent cations like magnesium, Mg2 plus, Ca2 plus. So, EDTA can also be used to disrupt the cell walls of gram-negative bacteria. So sometimes the components of the cell walls are held together, or the chains of the components are held together by these divalent cations. So if one uses EDTA, what will happen is this will chelate the Mg2 plus or Ca2 plus and uh, disrupt the cell wall integrity. And then definitely you have to later remove EDTA, because what happens is uh, in case of enzyme, because after lysis you need to, there can be other subsequent downstream processes where you need to use certain enzymes, etc. Now, enzymes, as we saw in the previous class, it requires cofactors to perform their functions, right? So, if certain cofactors can be metallic ions, and if EDTA is still present in your sample, then it will degrade or hamper the functioning of those enzymes. Okay. So, this has to be dialyzed away, or mainly by dialysis, what you do is you remove this or lower the concentration to negligible amounts. Okay. Then there's freeze thaw cell lysis. Freeze is some freezing. It's evident that we let ice crystals form. Now, what happens is when ice crystals are formed, uh, right, the volume, uh, because of its less low density, ice floats on water, right? Why? When because it has gaps inside, okay, due to its arrangement, due to the molecular arrangement in ice. So what happens is water actually expands upon freezing. Okay. Now, when a cell, particular cell or certain cells or group of cells are, you know, uh, resuspended and then in an aqueous medium, and then we are letting that particular sample freeze, then what will happen is ice crystals will start to form. And when water is freezing, it will expand, thereby disrupting the cellular structure or and letting the cellular contents out. But again, when you thaw the sample, again you are like, if you go through multiple cycles of this freeze and thawing, means letting the samples melt. Okay. So freeze and thaw cycle will let you will not let the cells to, come to an equilibrium condition, or uh, like take a break in between to really gather up its components, or to uh, recover from the shock of this formation of ice crystals. Okay, so that's how the whole cell is lysed. But then thaw cell lysis, this method is, it has to be, you should be going through several cycles of this to rupture the cells. And it is gentle, definitely it's gentle, but then as compared to, chemical, enzymatic lysis and sonication, this has to go through multiple rounds, and it can be ineffective if we don't really uh, like properly perform this particular freezing and thawing cycles without any other treatment or lighting method. Then it is gentle, and it won't generate any heat because we are letting ice crystals form, but it's definitely very time consuming. So for lysis, what people do is, ideally, there can be different combinations of lysis. Suppose uh, before sonication is definitely the most go-to thing before, separating out the components of a sample by centrifugation. So, people can first simply grind the sample, raw sample, in a suitable buffer, and then sonicate, or the sample can be enzymatically lysed and chemically lysed and then sonicated before further downstream processes. So different combinations of these lytic methods can be used to break open your cell. Okay. Cell of interest. Now, this slide shows the phenol extraction of DNA. Here we are talking about the genomic DNA extraction. So after we have lysed the cells, now what will happen is, people use phenol. Only phenol extraction can be there. There can be phenol chloroform extraction of DNA, or there can be spin columns having silica columns attached to these kinds of columns where there's a filter made up of silica. These are called spin columns, which can also be used to separate out or purify your genomic DNA or plasmid DNA, depending on what's your requirement is. Uh, So here, what happens in phenol extraction of DNA? You have lysed your cells. The cell is there in an aqueous soup, in a aqueous suspension. Now, when phenol is added to the aqueous soup containing proteins and DNA to be purified, okay. Now, phenol and water, they do not form a homogeneous mixture, and they are immiscible, and also water is less dense than phenol. All right. So what we can say is, see this pinkish part, and I don't know if it's clear, but there's a very transparent upper aqueous phase. So these two distinct phases, namely the upper aqueous phase and the lower phenol phase, or this is also called, organic phase, or uh, the upper aqueous phase is also called as polar phase. Okay. Now, phenol being denser out of the two, then it tends to sink to the bottom. And what will happen is, I showed you the vortex machine, right? This same tube, the small tube, can be put on that black area and vibrated at a certain speed. And then what will happen is, we let these two layers mix together. Okay. Definitely it is immiscible. So what will happen is, phenol, this will form droplets into the aqueous layer. So what will happen is, uh, because we are interested in getting the DNA, what will happen is, uh, proteins have both hydrophobic and hydrophilic moieties, like water loving as well as water hating or water fearing uh, groups in an amino acid, right? Because it has oxygen, which is water loving, it has hydrogen, which is water loving, nitrogen, which amide group, which is water loving. Okay, but then there's carbon chains or alkyl groups, etc. Okay, which actually tend to stay away from water. All right. So what will happen is, now when we are trying to mix these two phases, proteins, DNA, everything is there in this whole solution. Now proteins, again, they tend to see, what happens is, suppose this is the protein structure, DNA is also there. Definitely DNA is polar because it has all the polar residues, it is negatively charged due to phosphate groups attached to it. Okay, so this will be interacting with the aqueous layer or aqueous phase. Okay, now protein, suppose this is the structure of the protein. Definitely the polar residues, or which are the water loving ones, will tend to stay in the water area, will tend to interact with the water. whereas the ones which are water hating or water fearing or basically hydrophobic residues will tend to stay inside, curled up inside kind of. Okay. So this is how the ideal structure should be. Now what happens is when we are adding phenol, it is an organic layer. So which helps, which is actually more amenable for interaction with the less polar residues or hydrophobic residues. Okay. Now, due to the presence of phenol, these green residues, suppose the residues present in this green area will tend to interact with the uh, outer non-polar or less polar organic environment. Also, when phenol is present or organic environment is present outside, these polar residues will tend to move away from the organic layer. Okay. So now these polar residues shown by the blue area, these are getting inside, and the hydrophobic residues, these green ones, are flipped outside. Okay. Now proteins, the function of a protein is defined by its structure. Okay. Now if all the it's this is the structure, suppose, and then it is getting totally flipped out, the conformation, the configuration, everything is changing, suppose, then ideally it is denaturing the proteins. And the structure is completely disheveled, and what will happen is it won't be able to perform its performance function. The protein won't be able to perform its function, and so the proteins are denatured completely. But then DNA is very happy. Okay, DNA is in its aqueous phase interacting with water. So this is what happens. Now, because when we are vortexing, we are letting all these macromolecules interact with both hydrophilic and hydrophobic phases, right? And the proteins are flipped out, denatured, and that particular proteins in its flipped out configuration, flipped out means we are referring to the flipped out hydrophobic residues. Okay, this moves to the less polar part, and DNA keeps on interacting with the aqueous phase. So what we are talking about is proteins flip inside out. Green is outside, blue comes inside, because the less polar residues, which are usually hidden inside, now want to interact with the less polar or organic phenol phase. Okay. And polar residues on the outside of the proteins made them soluble in water here. Okay. Now the phenol induced folding changes have forced the phenol favoring residues to move towards the outside part because they will be interacting with phenol. So the proteins are now more soluble in phenol rather than water or aqueous phase. So what is happening? Some very polar residues may flip to the inside of the globular protein to be shielded from the unsuitable solvent. Globular is the structure, basically the tertiary or 3-dimensional structure of a protein, which actually renders its function. Okay. In short, the proteins are permanently denatured by the new solvent, here in this case phenol, uh, which is provided by the phenol. Now, we only saw phenol extraction of DNA. Next is phenol chloroform. Phenol is performing the same function, but what happens is sometimes you will get like here we saw two phases, right? Aqueous phase and phenol phase. Now, sometimes here at this particular the line between the surface between the two phases is called an interface. Sometimes it isn't as distinct as we just draw it out or schematically show it. This is not distinct practically. Okay, many times. So as to remove that fuzziness or to remove that fuzzy interface, chloroform is added. Okay. So what happens in phenol chloroform extraction is buffer saturated phenol is used, chloroform is used, usually like in a 50-50 ratio for DNA purification, and the ratios can change again with what type of molecule do you want to purify or what yeah. Sometimes this is also again another chemical isomer alcohol which is optional to use. This is used as an anti-foaming agent. Okay. Now again, buffer saturated phenol has higher density than water. What happens is it is not like very heavy than water. So what happens is if your aqueous phase has certain amounts of other kinds of solutes or salts, so what will happen that the water will also become dense. Okay. Now sometimes uh, what happens is because the density of aqueous phase increases, then we see a phase inversion. In the previous slides, we saw the transparent phase at the top, followed by the pink or phenol phase at the bottom. But if in case if somehow the aqueous phase's density increases, because the difference between phenol phase and water phase, difference in density of phenol and water is not that much, what will happen is if salts or other kinds of solvents, other solutes are present, then it can increase the density of the aqueous phase, thereby inverting the phase. Now your aqueous phase will go down, it will be transparent at the bottom and the pink phase will come out. But then what happens is, what are you interested in? We are interested in taking out the DNA. Now your DNA has gone inside. So then it doesn't make sense. What ideally was happening is, when the DNA was in aqueous phase, proteins and other like which we are not interested in, those biomolecules are in the organic phase. So what we do is we take out the aqueous phase very carefully, so as we don't allow the intermixing of the two phases, so that we only get the DNA containing phase, aqueous part. But if the aqueous phase only is getting at the bottom, then how will you really take it out? One way is there, like you can put a hole at the bottom of the tube and let the aqueous phase flow up. That is one way people sometimes try out, but ideally this should not be the case. Okay, phase inversion during extraction, because if the tube, then there's chances of contamination via the tube, that you are, if you are making holes, then again that has to be very properly like, done under sterile conditions. Okay, so it's better to keep the aqueous phase at the top and phenol phase at the bottom. For this to happen, chloroform is used, which is quite dense than water. As compared to water, it is significantly denser. So it keeps the phenol phase at the bottom. And chloroform again is an organic uh, thing which helps to precipitate out the proteins, or which helps to pull the proteins out of that DNA protein whole component, whole mixture. Okay. Now, chloroform adds to the organic phase and increases the overall density, thereby preventing phase inversion. Okay. Chloroform also has another function which helps to reduce the interface that I was talking about, the surface between the two phases. Sometimes it becomes fuzzy, uh, and this fuzzy border can be due to, some molecules which can't really decide where they want to go. Okay, like these can be due to partially denatured proteins which have all the polar residues inside and all the hydrophobic residues outside. That will happily go into the organic phase, but when in cases where some of the polar residues are inside and some are outside, then in that case, like that particular, just imagine the whole structure, then what will happen is it will tend to stay at the middle part, like not deciding, like not able to really decide which phase to go into. Okay, then some DNA molecules can also happen to be present in the interface, because the pH can, determine the stability and the charge on certain residues on DNA, and yeah, partially denatured DNA binding proteins. Now if DNA is bound by certain proteins and those proteins are partially denatured and the proteins interact very nicely with DNA, then so DNA bound proteins can also remain at that interface, not really able to decide which phase to move into. So what happens is, pipetting of some of the material while removing aqueous phase. If the aqueous phase is at the top and you try to pipette it out, then if by you have to be very careful. Even the pipette tip shouldn't be touching that phenol phase. Like by your naked eyes only, it's possible to very nicely visible the two phases, the two distinct phases are visible. Sometimes the fuzziness is there, so chloroform is added when you have very distinct separate layers. And when you try to pipe it out the aqueous phase, you should never let the tip of the pipette, as in the tip of the micro tip, to touch that phenol phase, because then you will get denatured proteins and other contaminants with your DNA sample. Okay. Again, there should be a balance. Like you shouldn't be too cautious while pipetting out, but then you are like, okay, Suppose 500 microliters of aqueous phase is there, and you are with the fear that, okay, I might contaminate my sample, I'm taking out only 100 microliters. Then it will drastically uh, lessen your concentration of the DNA that you will get finally. Okay, and which might be inconsequential to most of the experiments that you will be performing. Okay. Now we talked about phenol chloroform. So one way, some of the precautions that we need to keep in mind is we should avoid the oxidation of phenol. So we know that phenol, upon oxidation, forms a molecule called quinone. Okay, like another oxygen gets added. So oxidation of the phenol produces pink or brown oxidation products, and these can introduce cuts in the DNA and degrade RNA as well. Okay. In case we are interested in taking out RNA, then it is harmful for the RNA as well as it is harmful for the DNA, wherein it can produce random cuts on the DNA strand that we don't want. We want our whole genomic DNA to be isolated as it is, and then we want to perform further downstream functions. But first of all, to take the whole genomic DNA out, we have to be careful so that we don't lyse that particular DNA, or we don't fragment it into like fragment it down into further smaller fragments. Okay, at the level of lysis and just purification. So most commercial phenol solutions contain an antioxidant to inhibit this oxidation, and phenol buffered at an acidic pH has been shown to be resistant to oxidation. Okay. So it's a good practice to move a portion of the buffered saturated phenol. These again uh, are usually this buffer saturated phenol comes in brown bottles, to a clear bottle or tube to inspect it before extraction. Okay. Before using this kind of whatever phenol you want to use, you should inspect it for any oxidation that might have taken place. In case there's oxidation, you see the pinkish tinge or brownish colors, then you shouldn't be using that phenol for DNA extraction. Okay. Now, some of the advantages: phenol chloroform extractions are amazingly efficient, and less than 1% of the average protein have been found to remain in the aqueous phase after the first extraction has come to equilibrium. So also, like when we were talking about adding phenol and then vortexing it to create droplets or to mix the two phases. Okay, so that is done some 2 to 3 times, because many times there might be what we were talking about. Suppose some of the proteins haven't fully denatured, so they will be present in the aqueous phase, but or some are partially denatured. So then what will happen is, if we expose them to multiple levels of vortexing using phenol and aqueous phase uh, layers, like using like by exposing them to both phenol and aqueous phases, then subsequently they will tend to move to the organic phase only. Okay. So after that has come to equilibrium. Now this equilibrium is attained. What we are talking about is equilibrium between the two phases, when more surface area is there between the two phases. Two phases, aqueous phase and organic phase or phenol phase. So when the surface area between the two phases is more, then this equilibrium is attained faster. Okay. So how can we really increase that surface area? So we can increase it by what we were talking about, by mixing the two phases and vortexing the two phases, so that we can create emulsions, like small spherical emulsions. Now sphere is the larger surface area, right? So this equilibrium can be reached at a faster rate. Now, suppose what we were now, we started off with purifying genomic DNA. Okay. This is a very large piece of DNA. So what happens is, by purifying very large DNA like genomic DNA, we have to take care that we mix the sample very gently and perform each extraction for much longer, because we should always, if some kind of harshness is there, these genomic DNA, which are long stretches, these tend to get sheared, or tend to get fragmented. We don't really want that. We want our whole intact genomic DNA to be isolated or purified from the cells. Okay. Now, okay. Now you have purified your genomic DNA. Now that genomic DNA has to be put into a suitable carrier or a vector. Okay. So what happens is all these things here, like how to really clone into a suitable vector and all. I think that's in the next set of lecture videos, which we will be discussing in the next week again. But very briefly, if we go through it, like after getting the extracted genomic DNA, we try to add it or uh, join it into a suitable vector to prepare the genomic library. And the suitable vector can be selected based on what the size of our fragment is. Okay. And how much the vector can really take it. Okay. What is the carrying capacity of the vector? The size of average fragment can be calculated and accordingly a suitable vector can be chosen. In the case of fragment generated by restriction enzyme, which the molecular cutters we were referring to earlier, these carriers or vectors can be digested with the same enzyme and put for ligation or joining to get a clone. Okay. In the case of mechanical shearing mediated fragment generation, putting these fragments needs additional effort. Definitely. Okay. If we don't really know that, okay, what are the types of ends that we have generated of that particular DNA fragments, and then how are we going to really ligate it to a vector? If we know what kinds of ends are there, uh, suppose A type is complementary to suppose B type at end one, and at end two C type is complementary to D type. So if we ideally produce these ends using the restriction enzymes which have designated cut sites, okay, that is a good way to good approach to fragment your genomic DNA. But then if we randomly fragment it by mechanical lysis method or mechanical shearing method, what might happen is random fragments will be generated. Now you're definitely when you don't have a complementary region to B, suppose there's C present, but ideally A should have been there, then ligating it will be an additional challenge or additional efforts have to be put in to really ligate such ends wherein no complementarity is available or like the matching isn't proper. Okay. These everything this again we there will be a lot of details in these cases in this part in the next week. Okay. Ah. Now you have cloned the thing. Now what transformation basically refers to, you are putting the recombinant plasmid recombinant vector into an organism of your interest, like there we saw like uh, you can put it into bacterial cells. For that, the very first example of genetically modified organism, the plant cells we were talking about, coding for an insecticide uh, molecule. Okay. So those have to be really put, transformation is basically putting a foreign genetic material into an organism of interest. Okay. Now transformation to get colonies of bacteria, suppose we are talking about. Post ligation, clones are transferred to the suitable host to get colonies, and a suitable host can be bacterial strain or yeast, and different methods of delivering clones into the host cell will be discussed in the subsequent lectures. Okay. So these show, suppose if the carrier is plasmid, if the carrier is phage lambda, which is a bacteriophage, um, again, this is also a virus. This is bacterial artificial chromosome, yeast artificial chromosomes. So depending on the insert size, what is the maximum size of the insert DNA that or insert of the like DNA sequence that can be inserted into each vector, it depends on what is the carrying capacity. So for plasmids, it can take up to 50 megabases long DNA. Okay. Then these viruses can take up to this much. So yeast artificial chromosomes have the highest carrying capacity out of these, which can take up to 250 to 2000 megabases. Okay. So which is quite a huge number. But ideally in day to day, in experiments in laboratories, plasmids, phage lambda, bacteriophages are good enough choices, good enough candidates uh, for usage as vectors. Okay. Now we saw that how we can prepare genomic library. The other thing is when we have the transcriptome or the RNA content or RNA transcripts available to us, what to do with those transcripts. When we don't really know what the gene sequence is, what the genetic sequence is, Then what to do is, first, definitely if you have the RNA, then you have to isolate that particular RNA. So again, from your tissue of interest, suppose, or cell of interest, you have to isolate the RNA. You have to break open the cells definitely, and then here this the whole thing has been shown with respect to eukaryotic RNA. Okay, mRNA. Look, in case of eukaryotes, it has polyadenylated tail, right? Poly A residues at the 3' end. So based on that only, you can ideally use like thymine residues attached to certain beads which are there. See these small white colored spheres. Okay, so these beads have uh, thymine residues attached to it. Now your mRNA is having poly A tail at its 3' end. So ideally, due to complementarity, these beads will have poly A tail attached mRNA to it, and then you will ideally be treating it with different kinds of buffers so that the other biomolecules are washed away and your RNA of interest is attached to the beads. And then there's elution buffer. Elution buffer has anything which will be eluting out your sample of interest and decreasing the interaction of your sample of interest with the beads. Okay. So, somehow like we will be like what to say, decrease the interaction between the thymine residues present on the beads and the poly residues present at the 3' end of the RNA, and then get that particular RNA, and then further use it for downstream processes. Now, after you have RNA, then what you do is you reverse transcribe it to form DNA. Because see, RNA, because of its stability issues, and it's prone to degradation because RNA is enzyme which degrade RNA are present everywhere in the environment. Okay. So yeah, you isolate and collect mRNA as we saw earlier, and then you reverse transcribe it. This is a basic protocol workflow for forming complementary DNA library. You reverse transcribe it to get DNA, which is much more stable and can be, manipulated, and we can be a little we don't have to be very gentle and very cautious as we have to be in case of RNA, but definitely we have to be cautiously performing the experiments. But uh, with respect to RNA, we need not be that much cautious for about the degradation because DNA is very much stable as compared to RNA. Okay. After you get your DNA, again, what we saw with genomic DNA, what we were doing, it will be the same step. Like you are actually inserting this particular DNA into a suitable vector, and these vectors are then uh, put into, or these recombinant vectors are then put into cells of our interest or organisms of our interest, and then let them grow. And then when we have multiple copies of these recombinant vectors that we have put inside these black ones, now these black ones are inside the cells. Now what we need is we need to check that okay, whatever we have produced, does it really have the sequence of our interest or not? So what we have to do, we can't really do it in this way. We have to take it out again out of the cells. So you isolate these vectors or plasmids and purify the DNA again, and then you sequence it. Okay. So isolation of mRNA, then preparing complementary DNA fragment, then cloning it into suitable vector system, transformation in suitable host, and then just to finally confirm that okay, our RNA has indeed gotten into, or rather the complementary DNA has indeed gotten into the organism of our interest or in the recombinant vector. We should always send or give the sequence the samples, so sequence the plasmids, so that we can see that the sequence of our interest is present in that recombinant plasmid or not. This should also be the case for genomic DNA also. When we are ligating the genomic DNA into a suitable vector, we should ideally be performing sequencing again so as to confirm it finally. Definitely, there are other confirmation methods on gels also. Suppose you have a vector of 3 kb size and you are inserting something as long as suppose 500 bases long gene, uh, like gene sequence inside your 3 kb plasmid. Okay, 3 kb vector. Now, that recombinant plasmid, when you run it on agarose gel is basically a molecular sieve kind of stuff, which wherein the DNA moves from positive to negative, negative to positive end, because uh, at the negative end DNA is put, and now DNA being negatively charged, it's repelled by the negative end, and it moves towards the positively charged end, and like positively charged uh, electrode, and then what happens is it gets separated based on its size. Now, if that insert hasn't gotten into a 3 kb long vector, then it should run at the level like at 3 kb level only. But if the 500 base long insert has actually recombined with the vector, then it should run upper at upper levels, because being heavy, it will run slower as compared to the vector which doesn't have the insert. Okay. So this is another way to check. Agarose electrophoresis is also another way to check if your insert is there or not. Okay. So complementary DNA is an extremely useful tool which has been gifted to molecular biologists actually from RNA viruses, which use this process of reverse transcription uh, to sustain their life. So reverse transcriptase enzyme has been ideally uh, like rather gotten from them. Okay, people have used it, like taken the idea from the viral mechanism, and then we use it for our own purpose. Okay, so RNA viruses use this mRNA dependent DNA polymerase. mRNA dependent DNA polymerase. Reverse transcriptase is basically polymerase enzyme which uses mRNA as a template and adds DNA nucleotides, okay, to form complementary DNA. RNA viruses use this reverse transcriptase enzyme to convert their single-stranded genome into dsDNA or double-stranded DNA for integration in their host's genome. Okay, but in for the laboratory purposes, it can be harnessed to make cDNA or complementary DNA copies from mRNA templates. That means that reverse transcriptase can be used to synthesize a cDNA copy of every mRNA that is present in an mRNA extract, which can have numerous downstream applications like therapeutic applications, or for research purposes. Research means fundamental to answer fundamental questions. This can be of immense use. Okay, like synthesis of complementary DNA. Now, again, here everything is with respect to eukaryotic mRNAs, which have poly A tail at the 3' end. Now, what happens is there are two terms: first strand of complementary DNA and second strand of complementary DNA. What happens is, for first strand of complementary DNA, to synthesize first strand of complementary DNA, you are putting in mRNA molecules having poly A tails at the 3' end. And what happens is, because of its polyadenylated tail, there are oligo-DT primers. Okay, oligo means anything more than, suppose, like more than 3, 4, like multiple residues. Okay, which ideally range in the like 18 to 20 residues. So if oligo-DT primer, such 18 to 20, like all the T residues have been joined, and such primer will definitely go and bind to this 3' poly A tail, and then uh, what happens is, there are other nucleotides that are present in the reaction, that are added in the reaction mixture, and definitely this and the enzyme is added. Okay, reverse transcriptase enzyme is added along with a suitable buffer where the transcriptase might require other ions to perform its function, etc. So all such proper buffer condition is provided for reverse transcriptase to perform its function. And then what happens? There are nucleotides for this. Basically, it is a polymerase, right? So using this as a primer, this oligo-DT primer, it will add subsequent nucleotides from this pool, nucleotide pool, and add, like as in add the nucleotides, thereby forming the very first strand of complementary DNA. Now what will happen is you add RNA degrading enzymes like RNase H, which can remove the mRNA totally. Okay. Now you have only the first strand complementary DNA. Now for the second strand synthesis, what you ideally will be doing is now you put primers for the complementary DNA strand, and then this will be amplified. Okay. Like how it will be amplified? This is PCR or polymerase chain reaction, which we will be coming to in the subsequent slides. Okay. Now see, this is what we saw for the first strand. Now that poly mRNA has been degraded, primers have been added for second strand of cDNA synthesis, and then these can be further extracted for downstream processes. And there are adapter sequence, etc., which are actually uh, which can be synthesized, having different kinds of nucleotide sequence, etc., wherein which helps in cloning of certain fragments into different kinds of vectors. Okay. Now, what can be the advantages of forming complementary DNA over genomic DNA? So, no introns. Again, eukaryotic genes have interfering sequences or introns. Okay, in the messenger RNA, the mRNA that we get in eukaryotes, that is formed from so the after transcription we don't really directly get mRNA, right, in eukaryotes, wherein where you have actually what we get is hnRNA or heterogeneous RNA having both exons and introns, right? Interfering sequences between the coding exonic sequences, right? So that has to be removed. In case of genomic DNA, you will have all those sequences, and you have new genomic DNA along with the coding sequences that code for our uh, code for molecules of our interest, or performing the or in you or having its function in different kinds of metabolic pathways. There are many non-coding sequences that populate or crowd the genomic DNA a lot. So, but in case of complementary DNA, what is happening? We are making DNA from the mRNA, which has already its introns removed, right? So that is what is written here. We won't be having any interfering coding sequences or introns because these have been removed after mRNA synthesis. So complementary DNA, which is formed from mRNA, won't be having any introns. This means that a complementary DNA copy of a gene can be isolated as a single intron-free fragment. Prokaryotes don't have introns, so this is not a problem while working with bacterial genes. Okay, we can have more template. Okay, because there are multiple copies of mRNA for every copy in the gene, right? So more copies per cell of the gene of per cell of the sequence of interest. Okay, so you have more template from where more amount of or more number of cDNA can be formed, and there can be less background sequence. How? Because you only have the sequences that have been expressed as mRNA that will be present in a cDNA preparation, right? So there will be less background sequence compared to genomic DNA, which makes it less likely that the designed primers will bind non-specifically. What is being told here is that uh, again, there's this PCR, like earlier we were talking about oligo-DT primers, suppose. Now, if this could have been, we know that our mRNA of interest has number of polyadenylated residues, like polyadenylated tail at the 3' end. So oligo-DT primers will be binding specifically to that particular area. Now, if it would have been genomic DNA spanning a huge number of bases and nucleotides, there might be multiple regions where AAAA sequences are present, right? Multiple copies of A residues are present at different stretches. So the primers that we have designed, suppose oligo-DT primers we are talking about, those will bind to multiple areas of the whole genomic DNA stretch, and then uh, like whatever sequence of our interest we want, we might get some other sequence of interest as well. Okay, which we do not want for downstream processes. All right. So there will be less background sequence in case we are dealing with complementary DNA, because the mRNA has a specific sequence, and the primers that we are putting into the reaction mixture will be very much specific to our sequence of interest as compared to genomic DNA, which is a very long stretch of nucleotide sequence and can have multiple non-specific areas where our design primers can bind to and give non-specific products, okay, amplified products. Now, this for the synthesis of cDNA, there are two types, wherein one is homopolymer tailing. This is from the lecture video only. I am like just revising it because there's I think one or two questions in the assignment. So we are just going through it. So what happens here is in homopolymer tailing, this method exploits the presence of poly A tail at the 3' end here on an mRNA to synthesize the first DNA strand that we saw in the earlier slide, right? And then we degrade that mRNA using RNAs and then synthesize a second strand. Okay. So this is the schematic. So what happens in the first step, an oligo DT primer that we saw is used with mRNA as template to prepare the very first strand of DNA with the help of reverse transcriptase and DNTPs. Okay. DNTPs is deoxynucleotide residues. Okay. In case we need to suppose here we are talking about reverse transcription. If we were to talk about transcription, that is formation of DNA from, formation of RNA or, RNA from DNA, then what should have been the residues or nucleotides? It would be RNTPs or uh, ribonucleotides. Okay, ribonucleotide phosphate residues. All right. So DNTPs for DNA synthesis, RNTPs for RNA synthesis. Okay. In the second step, after the synthesis of first strand, terminal transferase, which is again an enzyme, is used to add C nucleotide on 3' of both the mRNA here. At this part, this is the mRNA and the newly synthesized strand of cDNA. Okay. At both the 3' ends, some C residues are added and this will form DNA RNA hybrid is loaded on an alkaline sucrose gradient. This step will hydrolyze RNA and allow the full recovery of cDNA. Now, what happens is, I think in the phenol chloroform extraction, we talked about DNA extraction in alkaline using alkaline medium. What happens is alkaline medium means because of its probably the OH group present there, it can be due to the presence of uh, like high amounts of OH minus in the medium. What happens is there's a reaction that makes makes the RNA this particular thing is controversial, but still there are possible explanations that can be given that why RNA is unstable in an alkaline medium. Okay, so this RNA, which is more stable in an acidic pH or acidic medium, so when you pass this RNA, like in the previous one, what we were doing, we were directly treating the reaction mixture with RNAs, which was selectively definitely degrading the RNA template, and then further cDNA synthesis was done using different set of primers complementary to the cDNA. Okay, now here we are not directly adding an enzyme. What we are doing is this DNA RNA hybrid is passed through a particular gradient, uh, which is alkaline in nature, having a high pH, and this step will hydrolyze RNA and allow the full recovery of cDNA. Hydrolyze means there's that phosphodiester linkage, right? So the OH minus will uh, have a is a nucleophile which will attack, and then the phosphodiester linkage will be broken between the bases, all right, between the nucleotides. Okay. Now what happens is once the RNA is hydrolyzed, you get the full cDNA here. Okay. This black one. And then what you add is oligo DG primer, DG deoxy, RG, right. Okay. Oligo DG primer is used with cDNA as template to prepare second strand of DNA with the help of reverse transcriptase and DNTase. Okay. This is homopolymer tailing. Another was preparation of, in case of radioactive probes, there's this terminal transferase method. Here also terminal transferase method was there, okay, which was actually transferring an enzyme, which was transferring these C residues at the 3' end of both mRNA and CDL. Okay. So what happens here in terminal transferase method also, it transfers a set of um nucleotide. Here again, because it's poly-A tail, what will happen is it will transfer T residues, DT residues, and then cDNA can be synthesized further. Okay. Now, your cDNA has been made, or cDNA library has been prepared, or the genomic library has been prepared when you don't know your gene of interest. Now, how will you really screen that, okay, my whatever library has been prepared, my gene, I want to now, out of this huge number of cells that have different types of different fragments or different genes of our interest, we need to now selectively select that particular cell having the gene of our interest out of so many number of cells, right? So one way is colony hybridization. What happens here is we basically replicate colonies onto a durable membrane, which can be a nitrocellulose membrane or nylon membrane, prior to hybridization with the labeled nucleic acid probe. Okay, what will happen is hybridization means letting a certain stretch of nucleotide bind, like establish complementarity with a gene of our interest, okay, sequence of our interest, all right? So if something, if a labeled probe, which is a short stretch of nucleotide sequence, okay, which is labeled, which can give a certain labeled, it can be labeled radioactively, it can be labeled uh, with a or tagged with a fluorophore. Like labeling can be of different, can be done with different kinds of residues. So if that probe is used and it is getting based on the regions of its complement, like based on the residues which are complementary to its own residues, the probe will be binding to that sequence of interest. So ideally, what we do is we design that probe, this particular labeled nucleic acid probe that we are talking about here. These probes are designed with in such a way that the residues in the probe are complementary to the sequences of our gene of interest. Okay. Now, this method is popularly used to identify colonies containing recombinant DNA should a suitable labeled probe be available. Okay. So what is happening here? Let's just briefly go through it. You have your bacterial colonies, suppose bacterial or yeast, whatever is there. Suppose here we have bacterial colonies that are growing on this particular medium. Okay. Now this is the membrane, which can be very, like, we have to be very careful about during these steps. We have to place it very carefully onto the bacterial colonies, which again here the bacterial colonies should be well separated from each other, right? Like it shouldn't be too, like how we were talking about the previous week, how there shouldn't be a smear, right? When we are plating the cells. All right, there should be distinct colonies visible, which can be suppose counted, or even if not counted, like it should be a decent number of colonies which are distinct from one another. All right. So this nylon membrane, if it's too crowded, overlapping colonies uh, won't make any sense, because this would give false positives or false negatives as well. So nylon membrane is put on top of this particular bacterial plate, and that membrane, what happens is you suppose this is a plate and this is your membrane, you are putting it here, you have colonies growing on the plate. Now what you do is, because that membrane is permeable kind of, now what you do is you put that membrane, this is your membrane, suppose you put it onto another medium, another plate which has a suitable medium for the bacterial colonies to grow. Now, because this is actually a membrane only, which is permeable, what will happen is the bacterial colonies can take up nutrients from the medium placed in the plate, and that they can grow, divide and grow. Okay. Now, invert membrane and lay over new agar surface allow colonies to regenerate on top of filter. Now, uh, you have to replicate this filter on a new agar. This is how the same plate is actually going over on this nylon membrane, and this membrane is then removed with attached colonies, and then certain treatments are given using solutions wherein you actually fix the DNA, denature, fix, wash the DNA, and then whatever probe we have designed, that probe is, this membrane is treated like with the probe. Now we have our DNA attached to it because after multiple rounds of treating with certain solutions, we have our DNA attached to it, and the probes will bind to that DNA of our interest where it can establish complementarity, right? So what will happen is, suppose here, what we say is, this is the complementary, like the probe has is giving signal at this area. Okay. Now, this particular area, this is the positive colony which can be further cultured and grown into, produced into multiple numbers, copies, and then pelleted down by centrifugation. Okay. So these are ideally containing our, these set of bacterial cells are containing the recombinant plasmids. Others do not contain the recombinant plasmids, or sometimes it also happens that like the cells have been able to take very less number of vectors, recombinant plasmids. Okay, so they aren't really producing enough amounts of gene of interest or coding for the molecules or suppose proteins of our interest. Okay, but this black one is the signal given by the probe, labeled probe, which can so again, here we have to be very careful while putting the linol membrane, because suppose one can do is like label it like 1, 2, 3, 4, 5, 6. There shouldn't be a symmetry, because if it's symmetrical, you might confuse between the nylon membranes, like between the bacterial colonies that are sticking to the nylon membrane. All right. So if it's sort of unsymmetrical, then you can really compare that, okay, this colony which was present on the left side or right side or top half or bottom half of the plate, then you can ideally pick the correct colony, positive colony, which gave the signal from the probe. So, and then that can be used for further downstream processes. So labeling is very important, and definitely carefully placing the membrane and labeling the membrane is also like maybe a cut mark at one side of the membrane to show that, okay, this is, suppose if a cut mark is made at this part of the membrane, then that means that this is the part where the very first colony was present. Okay, so suppose I have labeled this as 1, 2, 3, 4. Then if I made a cut mark at this area of the nylon membrane, this is like, okay, this is where the first colony was there, and then I can number it based on my convenience. Okay, it depends from one person to, like, it can vary from one person to another. This was colony hybridization. Next is screening by immunological methods. Same thing. What we are doing here, there's a master plate. We are transferring it onto, there we were transferring it onto a membrane. Here we have to transfer it onto a matrix. Cells are lysed and proteins are bound to the matrix. Okay. Here everything was lysed and only DNA was bound to the membrane. Here proteins are bound to the matrix. Okay. And then antibody, which is specific, which recognizes certain residues of our protein of interest or the bound protein, that primary, this whole thing is incubated with the primary antibody, and the matrix is treated with primary antibody. So suppose proteins of our interest are present only in this part. See, this V-shaped antibodies are binding here, and then secondary antibody, we incubate it again, treat it with secondary antibody, which are specific to the primary antibodies, not to the proteins of our interest. Okay, primary antibodies are specific, will specifically bind to the proteins of our interest, and secondary antibody will be specific to the primary antibody that we have added. All right. So again, there are wash steps in between, wherein these wash steps are always to wash away any non-specifically bound proteins or antibodies. And then secondary antibody usually is labeled, again, how we were doing in case of hybridization, the probe was labeled. So usually in case of secondary antibody, this is labeled. and this signal from where signal given by the secondary antibody can tell us, okay, which particular cells had our were expressing our proteins of interest. The remaining ones which did not give any signal, okay, from the secondary antibody, did not contain our protein of interest, but these particular cells, or suppose this particular colony of cells contained our protein of interest. Okay, so then we will be culturing this, but we like inoculate this cell and then culturing this and subsequently use it for our experiments. All right. Now I hope you guys know about like why are we using secondary antibody. This is basically to amplify the signal, because primary antibody, suppose only very vague example, very trivial example, which is suppose four proteins are there, four primary antibodies can bind to specific regions. Now, multiple primary antibodies can bind to only this area. But then if primary antibodies are multiply bound here and secondary antibody can recognize primary antibody, we are basically amplifying the signal. Okay. Only primary antibody can also be used, but then the signal from only primary antibody bound substrates can be low. So that particular signal can be amplified using secondary antibody, which can again be labeled with, fluorophore or some kind of chemical or enzyme, okay, which again further participates in a chemical reaction to give rise to a certain fluorescent compound or like luminescent molecule, like that, so that we get a signal and understand, okay, this is the part where our protein of interest is present. All right. Screening by enzymatic methods. This method is based on the ability of protein to exhibit an enzymatic activity. Again, this is not very specific, but can allow us to identify a class of protein where we know that it has that particular enzymatic activity. Okay. Now, isolation of gene. Once the position of a clone is known, it is extracted from the master plate and plasmid is isolated. Okay. Clone is further diluted to check the homogeneity of clone. Homogeneity of clone as in can be diluted and plated again also. The purity of the clone and presence of clone is further tested with a PCR using sequence specific primers. All right. PCR polymerase chain reaction. We will be coming to it in a very short while. And now, see, we talked about genomic DNA purification or isolation, and there's this plasmid DNA isolation. Plasmids are very small as compared to, like comparatively quite small as compared to large genomic DNA fragments. Okay, here, so plasmid DNA extraction, we will just briefly go through the steps because genomic DNA, what we looked at, plasmid DNA we have to be a bit careful with plasmid DNA extraction. Okay, and this is a little bit different from what we do in genomic DNA extraction. We can be a bit harsh with genomic DNA extraction. Again, definitely not too harsh with vortexing or mixing, because we might shear the DNA. But what happens here in plasmid DNA extraction is it is trickier because the plasmid DNA must be kept separate from the genomic DNA to avoid cross contamination. First, what you do is it definitely has to be lysis. Okay, but lysis here has it doesn't simply digest the cell wall as we were doing like with enzymes or chemicals or sonication, etc., as we were doing in case of genome DNA isolation, right? So what happens here is the lysis buffer in this case, the cells are the same. You have to put the cells into this buffer only. What happens here is it has sodium hydroxide and SDS, sodium rhodecyl sulfate, which is a detergent, which can completely denature plasmid and genomic DNA both. Okay. Basically separate DNA into single strands. This is crucial to perform quickly, because what happens is if we are late in doing this, or let the buffer or let the cells stay for too long in this buffer, what happens is excessive denaturing may result in an irreversibly denatured plasmid. So this denaturation that has happened, this complete denaturation of plasmid and genomic DNA, that is reversible if we are letting the solution stay for suppose 2 minutes or 3 minutes or something like that. There's a like specific protocol which we have to follow, the time bound protocol, which if we exceed that particular time, it has been tried and tested, and then that standardized protocol is present, wherein if you exceed that particular time, let it stay for longer in this particular uh, exposed to sodium hydroxide and detergent, which is SDS in this case, it can irreversibly denature the plasmid and it would be of no use later. Next is you added NaOH and SDS, then you have to neutralize the alkaline. This step is alkaline lysis. Okay, in case of plasmid DNA extraction, we do alkaline lysis followed by quick neutralization. What happens is we add potassium acetate to renature the plasmid. Okay, this is a very crucial step which actually in the first step, we were denaturing both plasmid and genomic DNA. But here we are interested in getting only the plasmid DNA, not the genomic DNA. So at this step only, neutralization is the step wherein you are able to separate the plasmid and the genomic DNA. All right. So plasmids are, because they are very small as compared to, like quite relatively small as compared to genomic DNA, they can easily reanneal or renature. Okay, thereby forming double-stranded DNA, but genomic DNA, being such large uh, sequences, they become actually those become entangled. So those take time to reanneal fully. So it tends to tangle so that the complementary strands remain separate. Okay. Now after every step, there's centrifugation. Okay. So after neutralization also, you have to centrifuge wherein genomic DNA, which is again bound to proteins, will come in the pellet, then plasmid DNA, which has, renatured and reannealed to form double-stranded DNA, remains soluble. Now, here we shouldn't, just after centrifugation, we shouldn't be vortexing it or, pipetting it out or something, or mix the sample, or do not, we shouldn't be disturbing the sample because the pellet that has come down, it might mix with the supernatant. Okay, the supernatant which has plasmid DNA and the pellet has genomic DNA, which we are not interested in this case when we want to just purify plasmid DNA. Okay. Yeah. So what might happen is, suppose if we are vortexing or mixing the sample vigorously, the genomic DNA is prone to shearing or fragmentation, so it can break down, and the small fragments can again become comparable to plasmid DNA sizes. And then because of its small size, like the plasmid DNA, the small fragments of genomic DNA can also reanneal and renature and move into the supernatant. Okay. Uh, Remain soluble. Uh, and thereby it can and go into solution with the plasmid, thereby contaminating our plasmid sample. Okay. So this shouldn't be done. We should be careful to not vortex or mix the sample vigorously. Then comes purification, wherein plasmid DNA in the supernatant can be precipitated with ethanol or cleaned up using phenol chloroform that we saw earlier, or using silica columns, using spin filter with silica columns. Okay, so here just a little bit of thing. DNA, we saw that DNA being polar, it tends to interact with water molecules. Now, if we talk about the force between two charged species. Those are Coulombic forces, right? If in a water molecule, I think if you are aware, there's dielectric constant of water. Okay, so the force between two charged species is inversely proportional to the dielectric constant of the medium. So the higher the dielectric constant of the medium, lower the force of attraction or force between two charges, right? So what will happen here is, because the dielectric constant of water is, I think, around 80, is around 80. So it's uh, too high. So the forces between the charged moieties, okay, are relatively lesser. Now what happens is, when we this precipitation, what is happening here, I'm trying to explain that when ethanol is given, ethanol has lower dielectric medium. So if the dielect, as in, is a medium with having low dielectric constant, if the dielectric constant, which is actually characteristic of the medium, if that dielectric constant is low, then because it's inversely proportional to the force between two charged species will come together. All right. So what happens here is, if we are adding ethanol, then we we not only add ethanol, we also add uh, sodium acetate. Okay. Suppose sodium acetate, lithium acetate, some kind of salt we add. So what happens is DNA being negatively charged and the salts having positively charged residues. That positively charged residues can DNA being a negatively charged moiety and the positively charged cations, as in the cations from the salt, can come closer because ethanol is there, which is allowing the two charged species to come closer, and the positively charged ions from the salt are actually what to say masking the negative charge on the DNA, thereby hindering its interaction with the medium. All right, and hence it precipitates out of the medium or solution, and we can then remove that particular solution or remove the ethanol or evaporate the ethanol, let the ethanol evaporate, and then finally we resuspend the pellet, which is our DNA of interest, which contains uh, the DNA of interest, and then use it for subsequent methods or downstream processing. Okay. Now, spin filter, what I was talking about, neutralization buffer in case of the spin columns contain some chaotropic salts, guanidine salts. Chaotropic agents are like chaos inducing agents, right? What they actually disrupt the interactions of the solute molecules, especially in this case DNA, with the water medium or aqueous phase. Okay. So the lysate can now, because it is because of the presence of salts, which ideally are more prone to interaction with the, which tend to interact more with the polar medium. What happens is the lysate, or in this case the DNA, can bind to the silica preferably now, but uh, can bind to the silica membrane that is used in the spin filter or spin column, and then the resulting DNA can be eluted out or precipitated out using ethanol here that we saw, which is letting the cations mask the negatively charged DNA and precipitate out of the solution, and also lowering its interaction with the solvent, aqueous solvent or polar solvent, thereby precipitating it out, and then it can be used for further molecular biology applications. Actually, these are the principles behind whatever you will be. If you have done practicals, then it's well and good, and if in future you're going to do these practicals, then these are the principles behind each of the practicals that you will be doing. Why are you using this particular buffer? Why are these steps there? Why do we have to be careful at some of the steps and not so careful with some other steps, like that? So these are the principles or theoretical basis of the practicals that you will be performing or you have already performed. Okay. Now, this particular alkaline lysis method is compatible with mammalian and other eukaryotic plasmids and some extra chromosomal DNA species as well. Plasmid DNA can typically be 3 to 5 kilobase long, and the specific origin of replication present will influence the plasmid copy number per cell. A typical high copy plasmid such as PUC or P blue script, these are like names of the plasmid, should be giving us this is purely for technical information. That's like once you start dealing with these on a day-to-day basis in laboratories, then you will get a hang of all these, these technical terms or this memory, like kind of like what are the names, PUC, P blue script, etc. But at this stage, this is just for your understanding that certain plasmids can be high copy number. That means it is produced by like replicated like giving rise to multiple number of copies, whereas some plasmids are there which are low copy number plasmids, like which are present in uh, like uh, like less numbers or less rounds of replication happens for those plasmids. Okay. To isolate high yields of plasmid DNA, use cultures in the late log phase or early stationary phase. Okay. Prepare cultures using fresh single colonies and fresh selection antibiotics at the right concentration for plasmid maintenance. Okay. Definitely, this antibiotic resistance is there, which maintains the plasmid selection. And it is important not to overgrow bacterial cultures, as this may result in genomic DNA contamination in the plasmid extract, because cells will start dying. If we overgrow, there will be competition for resources, etc. So it might hamper with the plasmid extraction or plasmid purification strategies. Okay. Now, DNA replication, like very briefly, I will go through it. That it is semi-conservative. It has a particular area from where it starts, and it can either be unidirectional when we talk about a particular only one direction, or in case of circular plasmids, it is bidirectional. It is semi-discontinuous. Leading strand 5' to 3'. Synthesized strand is the leading one, for which the template is 3' to 5'. Parent strand, all right. And lagging strand is the one which is synthesized in the 3' to 5' direction. Okay. Because again, because of the activity of the polymerase, or the way there's a loop formation as well, and it lags behind the leading strand. And for the DNA polymerase to actually start adding nucleotides, it needs a stepping stone kind of thing in the form of RNA primers. Okay. Again, this is due to the shape, the uh, whatever residues the polymerase is made up of, for for it to get into the active state. RNA primers are required, not DNA primers. Okay. Now, for DNA replication, these are the three steps. Initiation, which prepares DNA for synthesis. Elongation, this is the wherein you are actually adding the nucleotides. I mean, the polymerase is adding the nucleotides, and then finally, DNA synthesis stops. Okay. So DNA replication, these are some of the proteins, or rather enzymes involved in DNA replication. Helicases separates the two strands. DNA helix, it has to be separated. Primase, RNA primer synthesis. We saw in the earlier slide that DNA polymerase needs RNA primer to start building or adding or synthesizing the DNA strand. Then SSB protein, this is single stranded binding protein, which prevents re-annealing, because uh, it's not that the whole DNA gets uh, like separated into two strands. Certain stretch of DNA gets separated. So if strand is getting separated, so then there will be it has a tendency to recoil again. Okay, like again for like establish complementarity and recoil. So as to prevent that, once the two strands are separated, so as to prevent their re-annealing or recoiling, certain proteins are bound on the single strands, which prevent the separated region to re-anneal and form double-stranded, or single-stranded region to re-anneal and form double-stranded regions. DNA polymerase synthesizes new strand. There's tethering protein, which stabilizes the polymerase, and DNA ligase, which joins the uh, or creates phosphodiester linkages between the nucleotides. Okay. Especially like suppose there's DNA ligase wherein you have RNA primer. RNA primer has to be converted into DNA sequence, and then that DNA sequence with the remaining rest of the sequence has to be joined, right? So DNA ligase comes into picture in such cases. Now, now earlier we were talking about primer, PCR, like amplifying the gene of interest or amplifying the complementary DNA. So this was by polymerase chain reaction. So the basic steps of polymerase chain reaction is basically amplifying the set of DNA molecules when we have certain low quantities. What happens is initial denaturing, it will be denaturing, leads to melting of double-stranded DNA into single strands. This is there. The complementarity is broken, so you have two separate strands. Denaturation initiates following cycles. What happens is first you have the denaturation. Now multiple cycles of uh, synthesis, separation takes place. Okay. Initial denaturation is for the very first template molecules, template strands to separate. And then after that, what happens is each of the strand, the temperature, it happens usually at 95 to 98 degrees Celsius. Okay. Now, then the temperature is lowered down to uh, anneal specific primers, okay, at different regions of the DNA, and then you use certain thermostable DNA polymerases which can elongate the DNA chain using each of the template strand, okay, which is the elongation phase. Then then there's exponential amplification. After elongation, the target DNA strands are duplicated. That's why like this was why we were going through the DNA replication part. Like each strand, like each set of DNA, double-stranded DNA template can give rise to two more DNA templates. Okay, where each strand, each parent strand can form a daughter strand. Okay, and after elongation, the target DNA strands are duplicated. Exponential amplification allows the accumulation of specific PCR product. Okay, so this is the basic cycle wherein denaturation, which is usually at 95 to 98 degrees Celsius or greater than 90 degrees Celsius. Then comes annealing, where you lower the temperature for the primers to anneal to specific regions on your template DNA, okay, or template strand. Then you again elongate at little higher. First, you let it like bind at specific regions, bind as in anneal at specific regions, and then elongate it at slightly higher temperatures for it to form multiple copies of the DNA strands. Okay. Now this is a basic history of how PCR was developed. Okay. The very first DNA replication mechanism was first discovered, then different factors associated were discovered, then this thermostable DNA polymerase was discovered so that any other polymerase might, because it's a protein at high temperature, it can get denatured. But any thermostable polymerase like Thermus aquaticus lives in uh, at high temperature areas or high temperature regions. So in those regions, if uh, we purify or we use polymerase or enzymes from organisms thriving at such high temperatures, so in this range of temperatures, in this range of temperature at 95 to 98 degrees Celsius, we expect this polymerase to remain stable and perform its function. Okay. Then Mullis synthesized DNA oligoprobes for sickle cell anemia mutation. Then thermocycling was done to uh, form a small segment of clone gene. Then a patent was filed. Thermostable DNA polymerase was used for PCR. Okay, TAC was TAC was I do not was it is still used for PCR now. Still it is being used, and then instrument for PCR is called thermal cycler. Okay, and its reagents were developed and constantly being developed so as to decrease the amount of reagents to be added. There's master mix having all the polymerase already added along with its buffer, etc. So earlier, what we used to do was like add each component separately, like polymerase, nucleotides, etc., like all the DNTPs, adding one by one. So what might happen is that if one of the components is missing, then your PCR reaction won't work. Okay. But now, like things have been developed where the master mix already has the enzyme added, already has the buffer added, DNTP added, everything. You just have to add your template DNA, the primers, and that particular master mix having the enzyme already added, etc., and just set the reaction, okay, according to your specific reaction conditions. Now, primers again can depend on the primer length. We have to take care of the primer melting temperature, which usually ranges between 52 to 58 degrees Celsius. And the primer should be prepared in such a way that the difference between primers, as in here we are talking about forward and reverse primers, okay, should not be more than 2 degrees Celsius. So if the TM difference is more than that, actually 2 to 5 degrees Celsius is okay. But more than that, what might happen is it might have no like it non-specific, or it won't be able to bind properly only, okay, with the template. and then primer melting temperature and primer annealing temperature. Too high will produce insufficient primer template hybridization, definitely, because we are not letting the primer sequence bind to the sequence of interest. And if it's low enough, then that primer sequence, suppose if some of the nucleotide stretch is there at other regions of the template, then what might happen is the primer will bind non-specifically to that region as well. Okay. So GC content, again, the number of GCs in the primer as a percentage of the total basis should be 40 to 60%. It should be an ideal amount, because GC being having three hydrogen bonds, which forms three hydrogen bonds, it's a little stronger bond. So what might happen is, if the content is too high, then the primers will stay attached to the template strand, and if it is too low, then it might not give that much stringency in terms of annealing, okay, to the template strand. Then there's GC clamp. As GC forms a stronger bond than AT, the number of GC content at the 3' end of the primer should not be more than 3. Otherwise, yeah, this was what I was talking about, will result in non-specific tight binding at regions where G and C are abundant, because GC forms 3 hydrogen bonds and AT forms 2 hydrogen bonds. All right. So GC bond is stronger than AT bond. Also, sometimes what happens is, if between the primer sequences, there's a forward primer for one strand, one template strand, reverse primer for another template strand, right? So what happens is, these have to bind to the two different templates. Now what might happen is, the two primers can bind to each other. So that won't serve the purpose, because then there won't be any extension or addition of nucleotides, because there's no primer for it to specifically bind to a region, and then we need the amplification of our gene of interest. If the primer is not binding to that region of interest, then there's no point in synthesizing, as in there's no point in the PCR reaction at all. All right. So dimers should not be formed. Okay. Primer dimer is a structure forming double-stranded short stretches where the two forward and reverse primers bind to each other rather than binding to the template. And there might be repeats and runs. Repeats are consecutive occurrence of dinucleotide, whereas runs are continuous stretch of a single nucleotide. Maximum number of repeats and runs accepted is 4 dinucleotides, suppose 80, 80, 80, 80, and 4 base pairs respectively, A, T, 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 like that. Primer template homology. Primer should be designed in such a way that there should be no homology within the template other than the target site. Basically, to stop non-specific binding, okay, or amplification. Now, this is the formula for calculating the melting temperature, okay, for a primer length having less than 14 nucleotides, and for a primer length having more than 13 nucleotides. Okay, so this is the formula for this, wherein I think this will be, there's one assignment question where we will be using this particular formula. Okay, and then how would you be analyzing the PCR reaction? I talked about agarose gels earlier. Okay, so this is your template DNA, which is very heavy. An amplified DNA of interest will be a certain region in the template DNA, which will be definitely having lower stretch of nucleotides here. Okay, so here, see, there's always, we should always be running a reference, which we call as ladder or marker. So ladder can range having DNA fragments of different sizes, okay, which is usually company made. So what happens is, this is 500 base pair long, okay. This is 1000 bases long. This is 1500 bases long. This is 2000 bases long. This is 3000 bases long. So the template DNA is little lesser than 3000 bases, but the amplified, this is the whole DNA template, suppose, and the amplified DNA of interest will be a region in the template DNA, which is only around 1500 bases long. Okay, so the presence of this one, if it has, see, template DNA was there. Now multiple copies, multiple cycles of PCR reaction was there. So we are getting this amplified signal. If we had only the amplified DNA of interest with lesser number of PCR cycles, this strong signal or this particular signal due to This signal comes due to ETBR, right? Etherium bromide intercalating between the DNA sequences, double-stranded DNA. What happens is, so this will be lesser because of lesser amount of DNA produced, because we let the PCR reaction run for less number of cycles. Okay. And we see this is primer dimer. Primer dimer because primers are usually, around 18 to 20 or 15 to 20 nucleotides long. So these run much faster because of its very low size. So in case these are formed, these can run at such lower regions. Okay. Or it can sometimes go out of the gel also. Okay. Which when we can't really detect it, if primer dimers is formed, but then we do see the amplification of our DNA of interest, which shows that, okay, PCR reaction has worked fine. Now, what might be the applications of PCR? It can range from food science, uh, species identification, diagnostic therapeutics, criminal investigation, parental testing. Okay. So suppose in diagnostics, what happens is in case of an infectious disease, we need to detect that. Okay. So what happens is, suppose I'm having, suppose just a certain kind of disease wherein I do show the symptoms of it, and but my if it's at initial stage, okay, what will happen is some of the viral genome might be present in my some of the foreign genetic material might be there in my blood. Okay. So if the blood is taken out, suppose, and then the sample is like the symptom shows a viral certain bacterial pathogen or viral pathogen, then we see that, okay, the viral pathogens uh, sample is showing a match with my blood. So that means I have some kind of I have that specific virus inside me that is showing out that has made me sick, and I'm showing that those particular symptoms. Okay. So this is how we amplify a very small amount of genetic material that might be there, and then after amplifying the particular so if we already know the viral pathogen that has infected, or the genetic makeup of the viral pathogen, or suppose the bacterial pathogen that has infected, then what will happen is we will have specifically designed primers against that viral pathogens template. Now I will put from the sample that I have collected from my blood, then what will happen is I will expose that sample to, like, I will put that sample for PCR using primers, suppose use from bacterial pathogen and viral pathogen. Now, if it's a viral pathogen inside me, then the primers used or designed against the viral pathogens template will be amplified. And if it's a bacterial pathogen one, then the primers used against the bacterial template DNA will be amplified, thereby confirming that, okay, this is the bacterial pathogen I have to, like, that has made me sick. Okay. And suppose PCR is a very, very powerful technology to detect the diseases for genetic testing as well as for parental testing. Also, if somebody is prone to cancer, uh, this is a gene which has shown to be like BRCA gene, which has implications in cancer. So that BRCA gene can be PCR amplified to predict whether a particular person is predisposed to breast cancer or not using PCR technique. Okay. Also, this, I think, PCR, now RT-PCR had been a very, what to say, household term since the last 2 3 years during COVID, especially. So what used to happen was that uh, when, like, how where the COVID test performed, it was using specimen, that swab was taken, RNA was extracted and converted to cDNA. Okay. And that cDNA was PCR amplified. Okay. With SARS-CoV-2 specific primers. Now, if the specific primers are used and indeed the RNA uh, that was from SARS-CoV-2, then we would see the amplification. In case it's from some due to some other viral infection or there's nothing as such, then there won't be any amplification at all. Okay. So this was how PCR was used and RNA isolation was used for COVID testing. Okay. Or there would be RNA extraction free process also from saliva. Directly you can, again, use SARS-CoV-2 specific primers to PCR amplify the samples. PCR amplify the samples from like from cDNA and then interpret the results. Okay. So what used to happen was RT-PCR, this is PCR and reverse transcription. So either it can be one step wherein all the like reverse transcription from mRNA to cDNA is also taking place, and then after cDNA, like cDNA is getting amplified using PCR in one tube. Or it can be two-step also. First, you are using reverse transcription to form DNA, complementary DNA, and then you PCR amplify the cDNA obtained in this particular step. Okay, so it can be one-step RT-PCR or two-step RT-PCR. Okay, then it can be genetic testing also, wherein suppose we sequence the whole genome of the mother child, like potential mother and father of a child. Now, suppose there's this child B, wherein so a copy, this particular copy of gene 9 should be there in child, and particular copy of, suppose if 11 would have been there or 10 would have been there, then we can confirm that okay, this this person, this concerned person, is the biological father or mother of that particular child. Okay. There can be criminal investigations as well, especially from hair samples. Okay, what happens is sample of blood, hair root, or tissue left in the crime scene can be used to identify a person using PCR by comparing the DNA of the crime scene with that of the suspect or with DNA database of earlier convicts. Okay, see, this is the crime scene DNA profile, and see, these possible suspects have these DNA profiles. See, this doesn't match also, but this one, the suspect, this third number suspect's DNA profile matches well with the crime scene from the DNA obtained from the crime scene. So this is how the PCR profile, and it is a very sensitive as well as a very foolproof kind of technique to for criminal investigation. Like the tech, it's good to amplify the genetic material, how much ever sequences have been obtained or template have been there, different, if there are different sequences, then uh, different profiles will be there for different possible DNA sequences, and then the DNA profiles of each suspect can be matched with those. Okay. Okay, now let's go through the questions. These were the previous week's questions. Okay. Okay. So the release of DNA from DNA protein complexes. So DNA protein complexes can be many, but we can also talk about histone proteins. After lysing, the nucleus is achieved by digestion with which enzyme? Can anyone take a guess? Yes, good. Okay, option A, proteinase K. Because DNAs, we won't want that because it will digest the DNA. RNase H is, RNase H is for RNA and lipase is for lipids. Okay. Then the aqueous phase in phenol or phenol chloroform extraction contains what? If anyone can take a guess, or if anyone knows, like this is what we use for extraction, right? We talked about DNA extraction and all. So what we are interested in is the aqueous phase, right? Which contains DNA, which is option B. Proteins will be ideally in the phenol phase, okay, or organic phase. Okay. Next is the human chromosomes were digested with the eco-R1 restriction enzyme. The large fragments released could be ligated in the, so we are talking about human chromosomes, which is much, again, much larger than the E. coli ones. Okay. We talked about E. coli genomic DNA being around 4 megabases long, right? But human chromosomes is much, much larger than that. The large fragments released could be ligated in the dash vector. Okay. pre-digested with the same enzyme. So we looked at the carrying capacity of certain vectors, right? So the highest one was, does anyone remember? Yes, yeast artificial chromosome, option D. Okay. Now, so I should answer. Anyways, during isolation of genomic DNA, the nucleic acids are precipitated with what might be the thing? Any medium that is reducing like that has a reduced dielectric constant, thereby increasing the force between two charged particles, right? So what we were doing, we were doing ethanol precipitation, right? So what will be the option here? Yes, absolute alcohol. Okay. So next, which of the following organism has the smallest genome size? Does anyone know that? It will be, yes, good. It is mycoplasma. Okay. Now, the total RNA obtained or mRNA obtained from a particular cell type is known as, actually not only mRNA, it can be, we can also tell RNA also. Okay. What is that called? Yes, good. And then we talked about homopolymer tailing, right? So first it was DT, right? Oligo DT primer were being used. The second strand of DNA is synthesized with the dash primer. Does anyone remember? Okay. Yes, good. Option C, oligo DT. Now, cell division occurs in which phase of the cell cycle? M phase. Yeah, that is the M or mitotic phase. And when does DNA synthesis or DNA replication occurs? Yes, good. So cell division occurs in M phase or mitotic phase. Okay. So this is again a thing to be remembered. It was there in the lecture. The difference between the melting temperature between forward and reverse primer should not be more than, yes, 2 degrees Celsius. Actually, it can also be 5 degrees also okay. Sometimes it's okay. Okay. So yeah, from this option, it's 2 degrees Celsius. Now, in terminal transferase method of screening a genomic library, the probe is labeled with radioactive. It's a very direct question because poly tail is there. So what would be the probe? Radioactive probe? Yes, good. Okay. So which of the, again, this is from DNA replication, which of the following protein helps unwinding the DNA helix during DNA replication? So something that is breaking the helix will be good. Very good. Okay. So DNA ligase will be joining tethering proteins. Does anyone remember tethering proteins were used for? Yes. Stability to what? Like stabilizing what? Uh, of the newly synthesized, no, it actually the tethering proteins stabilize the polymerase, okay, which are kind of accessory proteins, okay, and the newly synthesized strand, or actually the single strands are stabilized by SSB protein or single stranded binding protein after the double stranded one has been separated into single stranded template strands, so that is stabilized by SSB protein, okay. Now, the role of, see, the role of the tethering proteins is to, now you have the answer. Yes. Okay, stabilizes the DNA polymers. I think three more questions. Actually, I have exceeded the time. Sorry for the head. The forward primer designed for a gene is this. The TM of the primer is, so this will be the formula that you have to use for this one. Okay. For less than 14 nucleotides, you can use this. So 4 degree into number of G and C plus 2 degrees into number of A and T. So if we go back, 4 degree into number of G and C. G and C 1, 2, 3, 4, 5, 6, 7, 8. 4, 8, 32 plus 2 into 1, 2, 3, 4, 5, 10. So 32 plus 10, it should be 42 degrees Celsius, right? Is it okay? Option A, 40. Now the role of SSB proteins is to, yes, good. Option C. And the primase synthesizes the primers required for DNA replication in cells, and the primers are made up of what? No, only RNA. Okay, option C. Because it needs just the nucleotides. Okay, and then it will subsequently add the like DNTPs. All right. Okay, then this was for today. Maybe I should stop presenting, but it's okay. So next Sunday we will be summarizing the lecture week 4 lecture videos and look at the assignments. And I will try to finish it within 5 o'clock. Today's Sunday, so you guys enjoy the weekend. All right. Okay then. See you in the next week. Goodbye. Thank you for joining.